It's 1938. In a drafty laboratory in Manchester in the United Kingdom, a young man, an undergraduate research assistant, hands a book of glass slides to the researcher who's been working at the best microscope in the lab for almost 8 hours now without a break. As this scientist stares carefully down the microscope, she patiently draws what she sees with a sturdy pencil. She sees thin organic structures that look like threads, light pink, uneven in length and width, and all jumbled up on top of each other. She thanks her assistant for the next batch of slides and she gets back to her work. She has a lot to do in order to put together all her research notes into a publication, or what in the research world is known as a paper. In the next year, her paper was published in what was then and still is the world's most prestigious research journal, Nature. A paper with the splashy title of Conchosalis Phase in the Life History of Porphyra umbilicalis. Okay, maybe not that splashy, but it is descriptive, as long as you know all the jargon. What does that all mean in practice? It means that she discovered that there are two very distinct phases to the life cycle of a type of seaweed species called Porphyra umbilicalis. In the phase she studied, the Conchosalis phase of the paper's title, this seaweed actually looks like a tiny incrustation on seashells. Very easy for a human to completely miss. But in the other phase, the one she didn't focus on in her paper, is the blade phase. This one is a lot harder to miss. Large brownish sheets that dry to a dark green out of water. You likely know them by their common name, nori, the seaweed found throughout Japanese cuisine and for decades now known around the world as a key constituent of sushi. Dr. Kathleen Mary Drew Baker, the scientist at the microscope, had been studying seaweed since the 1920s. She kick-started her research career in the United States at the University of Berkeley, but was dismissed in 1928 for the sin of getting married at a time when the university would not employ married women as researchers. But the University of Manchester, her alma mater, provided her a long-term home for her work. As far as I can tell from the writing about her, she was not attuned to or even particularly interested in the Japanese seaweed industry. Instead, she was simply passionate about seaweeds and studying them. My first experience in a scientific workplace was an assistant at the Seaweed Herbarium in the Natural History Museum in London, and I can attest that seaweeds are surprisingly beautiful and fascinating for their diversity. Different shapes, sizes, colours, life histories, and evolutionary lineages that we bundled together under the title of seaweed because they look a bit like plants and they grow in the sea. Nevertheless, immediately after she published her work, a fellow phycologist, that's a seaweed scientist, reached out to her, and he was very familiar with and interested in the Japanese seaweed industry. That was Professor Sagawa Sakichi. He worked at the University of Kyushu. Kyushu is just south of the largest Japanese island, Honshu, and it's a seaweed powerhouse. But the nori harvest, one of the few seaweeds that can be farmed, was really unpredictable. Japanese seaweed farmers had battled for some time with the problem of unpredictable harvests. Some years the harvest was bountiful, and other years it was meager. And in a stroke, Dr. Drew Baker gave Dr. Sakichi the information he needed to fix that. He got in touch with his colleagues at the local marine research station. Seaweeds, like all sexual reproducing organisms, have two phases to their life cycle. Humans, like all animals, have one life cycle phase that is vastly dominant, the type of human that you and I are. But there's another phase that is short-lived and is found just within the parent generation. We, as humans, call them sperm and eggs. For some animals, the sperm and eggs have a brief life outside of the parents, but they are always brief and have only one purpose, to find each other, fuse, and form a zygote, which grows into a baby. But for other organisms, it's not quite like that. For many seaweeds, the two phases can live independently for quite a while, and that's how it is for Porphyra. The blade phase, the one you can see and eat, is haploid. Like our sperm and eggs, they have just one copy of each chromosome. The blade makes its own little sex cells, which are just like the blade, haploid. They find each other and form little fruiting bodies that give off diploid spores, meaning they have two copies of the genome. The diploid spores land on shells and sprout to form the small conchosalis phase of thread-like structures. Inside the conchosalis is where meiosis happens, where the two genome copies exchange some information, and then during a specialized cell division process, the diploid cells produce haploid daughters. Those daughter cells are the spores that go on to grow new porphyra blades, the big sheets you can eat. The problem is, in a rough winter, the seashells would be washed out of the harbor where the seaweed was being grown, and so there would be no blade phase porphyra to harvest. the nori harvest would be ruined. Armed with this knowledge, Dr. Sakichi proposed a simple fix. Bring adult nori indoors as it's producing spores. Do this in a tank full of oyster shells, an ideal substrate for the Conchosaurus life cycle phase. Put some nets in the tanks for the baby porphyra blades to latch onto, and then once they've grown a little, bring them down outdoors to a carefully tended patch of coastal seawater to grow to adult size and harvest them. With this seemingly simple process in place, porphyros turn from a plant, well, actually a seaweed, they only superficially look like plants, into a crop. So what do I mean by that? What is a crop? A plant that is planted by humans to be harvested for some purpose. Seems straightforward, right? But in the case of porphyra, gives us that example of a not-quite crop, something that could be harvested, but that was, until the 1950s, elusive when it came to how exactly you make it grow reliably in a certain place at a certain time. Crucially, we didn't know how to control its reproduction. Controlling the reproduction of a food plant lets you essentially create food on demand, at will, predictably, and under your control. And it unlocks an ability even more powerful than that. Once you can control the reproduction of a plant, or an animal, or even a seaweed, you don't have control just over one generation, but all the generations to come. You can pick the offspring that you want to use for the next generation. You can select those with the traits you want. And now you get to decide not just where and when, but you have started to have control over the what, and bring the biological destiny of a whole species under your control. Welcome to Modified. I'm your host, Dr. Orlando Delange, ex-plant scientist and current science teacher. This podcast is my attempt to guide you through the science of GMOs and hopefully just enough of the history and the social context to give you a path forward in your own thinking. And if all else fails, I hope you'll enjoy some of the surprising stories and fascinating facts in the world of genetically modified plants. This is episode 6, What Makes a Plant a Crop? This episode will not attempt to chart the history of agriculture. That would require a whole podcast in itself. If you want to read a history of agriculture, there are plenty out there. One book I can particularly recommend is Hybrid by Noel Kingsbury. The book doesn't spend much time on the earliest origins of agriculture, which is a fascinating topic, but instead focuses on the 1500s onwards and how we ended up with the agricultural system still in use today. It was a huge inspiration for this podcast, and while definitely quite dense, it is still very readable. So instead of trying to give some sort of comprehensive history of agriculture, this episode is going to look at what a crop is to see what makes it different from a plant. The definition itself is the simple part, but we'll dig deeper with a few more case studies, expanding on the nori case study we started the episode with, to really unpack the implications of turning plants into crops. So, the definition: a crop plant is a plant grown for human purposes, most obviously for food, but it could also be for fuel or for fiber or for other things. For example, I live in Seattle, and just north in Skagit Valley is a vast multicolored blanket of tulips, which represent an important ornamental crop. So a crop is any plant that humans grow with a goal of harvesting and using it. So is that the same as domestication? No, but they are connected. Any plant can become a crop instantly if you start growing it for human use, even if the version of the plant you are harvesting is no different from the wild-growing version. Domestication is a change over time to make the crop more user-friendly for us, the human users. The two are connected and usually happen in tandem. As a plant starts to get grown by a lot of people, domestication will happen along the way. Domestication requires significant heritable change to make an organism suited to being grown or raised for human purposes. In plants, domestication generally involves larger harvestable organs, bigger fruits, leaves, or whatever it is that's harvested to use, and it often involves changes that make them easier to grow and harvest, such as straighter stems and fewer spines. Squashes, which is pumpkins and related crops, are a typical tale of domestication. Like any of the crop plants cultivated today, they were first domesticated thousands of years ago. About 8,000 years ago, it seems, in the case of squashes, in Central America. Like many crops, its history and its domestication can't be pinpointed in one exact place and time. It seems that many people were growing similar cultivars in different areas, domesticating bit by bit. Seeds moving around with people, cultivars of squash being hybridized and exchanged to become, over millennia, the more recognizable varieties we grow today. Yes, including pumpkins. Unlike many modern crops, the domestication journey was one of dramatic transformation, from a relatively small fruit with incredibly bitter flesh grown to be used as a storage vessel to a sweet, nutritious food crop. So with our definition of crops and domestication, let's look at another case study, another recently created crop, the sugar beet. It's 1812 and you're on your way to see a friend of yours who, like you, is a well-to-do member of Parisian society. He has invited you round for tea, but you're a little hesitant. You hate to take your tea without sugar, but you know that Emperor Napoleon banned all imports of sugar from the British Caribbean colonies. After all, you are at war against the British. But because of that, almost no one has sugar, and you're already dreading the bitter, unsweetened tea. Such a hardship. After you say hello to Gustave and take a seat in his drawing room, he updates you on the latest gossip from the court, and then he offers you a scoop of sugar with your tea. Shocked, you ask how he got it, and he laughs at your ignorance. It's Prussian beet sugar, of course. Perhaps a little more expensive than the usual cane sugar, but it tastes just the same. You take a sip from your tea, and you have to agree. Beetroot was domesticated in the Middle East thousands of years ago. So for thousands of years, the beet plant, Beta vulgaris, was grown as a food crop for its large edible roots. And for a similarly long time, it has been grown for its edible leaves, which we call chard. Like most other crops, the exact details of where and when it was domesticated, how its properties as a crop were discovered, will never be exactly known. We can only know rough areas and timelines. We cannot say who invented beetroot or chard as crops. But we can say who invented the sugar beet as a crop. The same plant as beetroot and chard took on a new life as a new crop, the sugar beet, in the early 19th century in Prussia, northern Germany, with a team of farmers and factory workers, all working under the direction of Franz Karl Achard. He had been the student of Andreas Marggraf, who in 1742 had carried out experiments to show that sugar can be extracted from beets, and that this substance is exactly the same as the sugar that is made from cane. Sugar cane is another of those ancient crops domesticated many millennia ago, this time in Asia and then spread around the world. Marggraf was a scientist and excited to be able to chemically describe sugar, specifically showing that the sugar cane and beets yield the same type of sugar, sucrose. But where Marggraf saw inquiry, Achard saw industry. He worked out how to extract the sugar at scale and turn it into a marketable product. And just as crucially, he used the nascent field of plant breeding to develop a variety of beet that has a higher sugar content. Plant breeding will be the focus of our next episode, but for now you can just imagine Achard doing what farmers have done for millennia, picking the best individuals to keep seed from and so iteratively improve the crop. But plant breeding was taking off around the time in the 18th and early 19th century, and was and still is about trying to do that in a very intentional way, and ideally as rapidly as possible, with the greatest changes achieved in as few generations as possible. Among the simple but powerful techniques used at this point was progeny testing, taking one promising individual plant and crossbreeding it with a deliberately wide and varied range of others to then study the offspring. Those diverse crosses will reveal a lot more about the potential of the plant than you would otherwise discover by letting it reproduce with itself or with similar plants. What we would say now is that it can reveal more of the underlying genetics, but in the early 19th century, neither the word nor the concept of genetics existed, and as we'll learn, the development of genetics as a field fundamentally transformed plant breeding. But plant breeding itself is older by a couple of hundred years. So Achard combined plant breeding with industrial process engineering to turn the beet into a new version of itself, a new crop, the sugar beet. Was he motivated by pure curiosity about what was possible? Was he motivated by hatred of the British who dominated the sugar trade? Or perhaps by hatred of slavery on which the British sugar trade relied? If you know, send me an email, I'd love to find out. But one way or another, he started a nascent sugar beet industry, one that would boom after 1811 when Napoleon banned imported sugar. Sadly, by this point, Achard was already destitute after his factory burned down in an accident. It had its ups and downs, but to this day, a good chunk of all sugar eaten around the world comes from beets, not sugarcane. In the US, most domestically grown sugar comes from beets. What this example illustrates is that a crop isn't just a plant grown for human use, but instead it captures all the ways that the humans use and reshape the plant and how it exists in the world. sugar beet, and Swiss chard come from the same species of plant with the same taxonomic name, Beta vulgaris, but they are different crops. Partly this is because selection and plant breeding have been used to make varieties of Beta vulgaris, or what we call cultivars, with thick sugary roots for sugar beet and succulent leaves for Swiss chard. But even more than that, even if there were not biological differences, there are socially constructed differences. The way you harvest is different, requiring different machinery and expertise, different systems for storage and transportation, and then they can be marketed in different ways, and of course used by the end consumer in different ways. The identity of any given crop is all of these things, biological and the social. Beets aren't actually unique at all in having many crops that all come from the same plant species. Did you know that Brussels sprouts, cabbage, kale, collard greens, cauliflower, and kohlrabi, among others, are all the same species, descended from the same ancient ancestor and made into a dizzying array of different crops? When the distinctions are more subtle, we might not call them different crops, but just different cultivars. For example, the maize grown to eat as sweet corn, the maize grown for popcorn, and the maize grown for corn syrup are distinct varieties that are not used interchangeably. And if you are the farmer or the processor, you might well think of them as different crops. So once you have a crop, how do you keep it? How do you stop it becoming something else? A crop must be constantly maintained. It exists alongside us and it develops and changes over time unless you actively stop that from happening. To maintain a crop, you need to maintain both its biological features and its social features. Let's start with the biological features. That is in some way easier. Imagine, essentially, what this means is preserving the DNA. And when it comes to plants, what that means in practice is seeds. Seeds have evolved to live outside of their parent plant by themselves in the world for some time. Just like the Conchosalis life stage of Porphyra, the seed is a separate phase in the life cycle of the plant. It has its own unique cell types and tissues. But its only goal, from an evolutionary perspective, is to survive long enough for good germination conditions to arise. And for all plants, at some point, if those good germination conditions don't appear, then the seed will die. So if you want to make sure that you don't lose your crop, you just keep planting every year, or at least very regularly, which makes sense to do anyway since you want the crop to be able to harvest and use it. So you just grow it in the same general area over years, which become decades, then centuries, and for some crops, even millennia of continuous cultivation in the same geographic area. And that is the way things have worked for most of human history in most of the world. Continuous cultivation over relatively restricted areas, with farmers in each keeping seed to plant at each cycle, with some selection, some trading or sharing within local markets. Well, what that does at the genetic level is create localized varieties of the crop. But unlike today, when crop varieties are things you can order from a catalogue, and that what you'll get is genetically uniform seeds so that you can grow a whole field of genetically uniform individuals. That requires a lot of careful techniques which are pretty similar to animal breeding, where you have defined breeding parents used to produce seed. Though whereas in animals excessive inbreeding is avoided, in plants repeat inbreeding is actually crucial to how plant breeding works. So crops as grown today tend to be grown in genetically uniform fields. The alternative, as practiced by farmers for millennia, is called land races. A land race is a crop variety that is genetically diverse, the inevitable consequence of allowing plants in and between fields to openly pollinate and therefore interbreed with each other. Land races often have lower yields than bred crop varieties. Because of their genetic diversity, they are more resilient. A disease that can infect some plants in the field probably won't be able to infect all of them. Remember back to those gene-to-gene interactions from episode 5? Even small genetic differences can have a big effect on plant disease resistance. So in summary, land races are the traditional way that crops are grown and how they are maintained through time. But since the 20th century, private entities and national governments around the world have invested in seed banks, generally focused on the preservation of crop seeds. You might have heard of the Svalbard Global Seed Vault, run by the Norwegian government and located on an Arctic island, using the natural cold to reduce the cost of running freezers and providing a fail-safe if the power goes out. Though climate change is sadly starting to imperil that strategy. Artificial cooling is a crucial part of how most seed banks extend the lives of the seeds in their collections, along with controlling humidity. Dry and cold conditions are best for preserving seeds. But even then, some seeds are hard to preserve long term, and need to be periodically germinated and grown into adult plants to produce new seed to put back into the vault. The global seed vault actually houses backup seeds that are subsets of the larger seed collections of other countries and institutions, hence the global part of the world. I actually spent a happy summer doing some lab assistant work at the Millennium Seed Bank, which is run by Kew Gardens and housed in the grounds of a beautiful botanic gardens in Wakehurst, West Sussex. It is not part of the main Kew site in West London. One of the most dramatic and notorious incidents in seed bank history is the preservation of the Institute for Plant Industry in St. Petersburg, Russia, during the siege of the city by Nazi forces in the Second World War. The scientists managing the seed bank were trapped along with everyone else, but never tried to eat any of the many edible crop seeds and even potato tubers that were stored in the seed vault, with 19 of the team of scientists dying of starvation during the siege. A really tragic episode that illustrates just how seriously the preservation of crop varieties is taken. I'd guess that at least 99.9% of humans live in societies with a farming-based food supply. And within those societies, there are so many social actors who care about crops and have a stake in them. As seed banks demonstrate, national governments and other political authorities care very deeply about crops, because maintaining a food supply, what we call food security, is just about the most fundamental responsibility they have. People cannot live without food, and in a farming-based food system, food doesn't just happen by itself, it needs constant work to produce and constant vigilance to maintain. And of course, farmers care about crops. Consumers care too, of course, not just because crops are what we eat to survive, but in every part of the world, food crops are not simply interchangeable food units. They are a source of identity, comfort, and communication of who you are, your traditions, and your ways of life. Putting it another way, they are deeply embedded in culture. But beyond the farmers, the political authorities, the consumers, there are the capitalists and everyone employed by them, the millions of people around the world who make a living out of the business of crops. They invest in the development of new farm machinery, which for hundreds of years has turned farming into an increasingly high-tech activity. They invest in the development of new chemicals to fertilize the soil and protect against pests. And yes, they invest in the development of new crop varieties, harnessing the science of genetics to produce combinations of genes that have never existed in nature. And there's also the social group of scientists, acting within all of them, taking money from governments, often with the explicit instruction to research questions of interest to the capitalist, but also based on the perceived needs and desires of the crop consumers. I think that a lot of scientists who got upset about anti-GMO sentiments see the argument as one between themselves and the crop consumers, when really all of these social actors, consumers, farmers, scientists, governments, owners of crop product companies, are all invested in the same thing, crops, but often do not have the same priorities or the same set of assumptions driving their beliefs and actions. Anyway, there was obviously a lot to unpack there, and that's what I'm going to try and do in Season 2 of this podcast if I ever get around to making it. But instead, for now, I want to end by just talking briefly about a new crop, or one that is going through a long, slow gestation period that it may never actually be birthed from. That crop is Kernza. Ker for kernel, and Ernza after Konza Prairie Biological Research Station, in turn derived from one of the names for the indigenous Core Nation where the state of Kansas gets its name. Most of the calories consumed globally are from cereals. That is the family of grain-producing plants, think rice, maize, and wheat, as well as others like sorghum, millet, rye, and oats. They all look roughly the same, tall grass plants with clusters of tiny flowers that turn into hard, starchy seeds called grains. Another thing they have in common is that they are annuals. That means they are grown from seed each year, they are harvested, and they die, with that cycle repeated annually. And that means at each cycle the soil ends up getting disturbed and nutrients depleted from it. Plants that can live more than a year, essentially indefinitely, are known as perennials. There are perennial crops. They include all the tree crops, but not just tree crops. Strawberries are an example of a non-tree crop grown as a perennial. Kernza is one of the several ongoing crops in development, attempting to create cereals that can be grown as perennial crops, reducing the need to add chemical fertilizers and to reduce soil erosion. Essentially, it should be more environmentally sustainable, and that should also reduce costs for farmers who don't need to buy fresh seed for each season and don't need to spend as much on fertilizer and other chemical products. Kernza grains can be used like wheat, but the starting species for Kernza is actually a wheat relative, not the same plant as bread wheat. Kernza is derived from intermediate wheatgrass, which is naturally a perennial, but it has low yields of small grains and lots of other features that make it less than ideal for farmers and consumers. In 1982, crop scientist Peggy Wagner selected intermediate wheatgrass as a promising candidate for a perennial cereal crop. She began work on breeding, but it didn't get too far. However, in 2003, the Land Institute in Kansas launched a national Kernza breeding program working with universities and with US government funding. Since then, the breeding effort has continued at a faster pace, and by now it is a commercial crop, but still a very tiny player when compared to the big hitters of US agriculture, partly because Kernza still only yields about a third of what you'd get from wheat. And also, just because it's new. As I hope I've made clear, crops aren't just interchangeable. Each crop has its own growing techniques, its associated technologies with expertise requirements and specific conditions, and it has to have a market of consumers who want to buy and eat it. All of those things need to be in place for a crop to be successful. But if the idea of new, more sustainable crops is appealing to you and you don't mind paying a premium for it, then go online and buy some Kernza flour and start baking away, or pick up a 6-pack of Kernza Pilsner from a collaboration between Patagonia and Dogfish Brewing. Kernza is a crop being fully developed in the modern era of plant breeding, using all the tools of genetics to speed up the process. That means tracking and predicting trait combinations using Mendelian genetics, and it means sequencing genomes and using computational tools to understand the genes in Kernza and what that can tell us about the less obvious biological features of the plant, and how that can drive plant breeding and improve farming techniques. In our next episode, we'll dip into the ways that genetics and plant breeding became intertwined in the 20th century, and how that became such big business in a world of billions of humans. Well done for listening and engaging with this rapid and erratic tour of what makes a plant a crop. Please do subscribe and tell a friend or leave a review. And if you have any questions or feedback, get in touch at orlando@modifiedpod.com. You can find the website with more info and show notes at www.modifiedpod.com. I'm still ironing out the details, but in January, in Seattle, I'm going to be doing a hands-on DNA extraction and science talk about genes and genomes. So if you live in Seattle and you want to get the details about that, and hopefully other upcoming live events, make sure to follow me on Blue Sky or Instagram. My Instagram handle is @SeattlePlantNerd, and my Blue Sky handle is @OrlandoDL. And you can find both of those at the podcast website, which once again is modifiedpod.com. And now I'll end this episode on my 5 take-home messages as always. Number 1. Crops are plants grown for human purposes. Number 2. Control over reproduction is a requirement to turn a plant into a crop. Number 3. Domestication is the significant modification of the biological properties of an organism to better suit human purposes. Domestication is an almost inevitable consequence of the transition from plant to crop, given enough times. These days, it can be done very rapidly, as exemplified by Kernza. Number 4. A crop is a biological and a social entity. Almost everyone in every society cares deeply about crops for a range of reasons, and sometimes goals and assumptions conflict between those groups. Number 5. For most of human history, crops were grown continuously in the same geographic area, producing distinct varieties that still have local genetic diversity. These are known as land races. That's about it for this episode. I hope you're listening again soon. Bye!